Now that you have a great understanding of Mendelian genetics, non-Mendelian genetics, linked genes, and how to analyze results of genetic crosses using chi-square analysis, we're going to go through a practice problem that puts all of that together. And we're going to use the results of crosses between different types of fruit flies. Fruit flies are really great subjects for genetic experiments because they have a lot of different traits, such as eye color, body color, wing shape, and so we can cross these. They create a lot of offspring, so we have a lot of data to look at to analyze the inheritance patterns in these organisms. So let's take a look at our first sample cross. Here we have true breeding bronze-eyed males crossed with true breeding red-eyed females. All of the F1 offspring have bronze eyes. If we cross the F1 flies, the data for the resulting F2 generation are shown in the table. So what can we actually learn from this data? Well, let's take a look. First of all, we know that the male is true breeding bronze-eyed. Because he's true breeding, it means that he's homozygous. And the female is also true breeding, meaning she's homozygous. Now, when this true breeding bronze-eyed male mates with the true breeding red-eyed female, all of the offspring are bronze-eyed. What that tells us is that the bronze-eyed trait is dominant and the red-eyed trait is recessive. So here we have a homozygous dominant male and a homozygous recessive female. So the offspring that result in that F1 generation are all heterozygous. And they would have one dominant gene and one recessive gene. Now, when we look at a cross between the F1 offspring, we would expect there to be a 3-to-1 ratio, bronze eyes to red eyes. And if we look, that's pretty much what we see, 3-to-1 ratio. There's here in our data table. So those results are expected. Now, if we take a closer look at the data, we can also see that the males and the females have about the same number. What that tells us is that this trait is known as autosomal, meaning that the gene for eye color is located on an autosomal chromosome rather than a sex chromosome. So from this information, we can see that bronze eyes are dominant over red eyes and that the trait is carried on an autosomal chromosome. Let's take a look at another cross. Here we have true breeding normal winged males crossed with true breeding stunted wing females. All of the F1 offspring have stunted wings, and when we do the F1 cross, the F2 fly results are in a table. So if we take a look here, we've got our normal wings crossed with our stunted wings, and all of the offspring have stunted wings, which means that stunted wings are dominant and normal wings are recessive. So our heterozygotes have one of each gene. Now again, we would see a 3-to-1 ratio, and that's exactly what we see. Stunted to normal is about 3-to-1. And our males and female ratio is pretty much equal. Therefore, this is another autosomal gene carried on one of those autosomal chromosomes. It's not sex-linked. So now let's beef it up a little bit and do a dihybrid cross. Let's take a look at both of these traits at the same time. So here we're going to take a true breeding bronze-eyed stunted wing male and cross him with a true breeding red-eyed normal winged female. Then the F1 offspring, we notice, have all bronze eyes with stunted wings. The F1 flies are going to be crossed with a true breeding red-eyed normal winged fly, and the results are shown in the table below. So let's first figure out how this cross works. So here we have a true breeding bronze-eyed stunted wing male. We know that both bronze eyes and stunted wings are the dominant trait, whereas the red eyes and normal wings are the recessive trait. So our F1 offspring are going to be heterozygous for both traits, and then we're going to cross the F1 offspring with homozygous recessive flies. And these are the results that we actually observe. Before we even look at these observed results, let's just make a prediction about what we would expect from our F1 cross with our red normal winged flies. So remember, our F1 flies are going to be heterozygous for both traits. And so when this fly packages its genes into gametes, we can have lots of different combinations. We can have both of the dominant genes get packaged into a gamete. We can have one dominant and one recessive. We can have this recessive with this dominant gene, and we could also get both recessive. That combination. Now, this female here is homozygous recessive for both traits, so the only type of eggs that she could make are eggs that carry both a recessive gene for the eye color and a recessive gene for our wing shape. And so when we look at the possible genotypes of the offspring, here are all of the possible genotypes. And then we can start to analyze, well, these ones would have bronze eyes with stunted wings. These ones would have bronze eyes with normal wings. These ones would have red eyes with stunted wings. And these ones would have red eyes and normal wings. And so what we see is we would have a 1 to 1 to 1 to 1 ratio. So thinking back to our data, we had 10,000 total offspring that were created. And so with a 1 to 1 to 1 ratio, we would expect to see 2,500 offspring with each of our possible phenotype combinations. But what we actually see is very different. And so when we look at this data, we can make some observations. Number one, we can actually do a chi-square analysis to prove that our hypothesis should be rejected. So let's do that first. So we're going to do our chi-square analysis. Here we have our equation up here. So we're just going to take the sum of all of our observed values minus our expected values squared over our expected values. So there is the value that you would get from the bronze stunted winged. Here is the value that we would get for our red normal wings. Add it to our values for the bronze and normal wings. And when you actually do this math, you're going to notice that your chi-square value is really, really large. Now, because we had 4 possible phenotypic combinations, our degree of freedom is going to be 3. We're going to use the 0.05 p-value, so our critical value is 7.82. Well, this number is so much bigger than our critical value. And therefore, we are going to reject our null hypothesis. So we hypothesized that these genes sorted independently. Well, now we know that they don't sort independently because we just proved it with our chi-square analysis. What that tells us is that the genes for eye color and wing shape are linked, meaning that they're on the same chromosome. And so what we can actually do is create a gene map. Now, we didn't really need to do the chi-square analysis. We could see that these observations, these observed results, are nowhere close to our expected values. Here are all of our recombinants. So this is where we would see some of that crossing over occur. And now if I take the total number of offspring, it's 10,000. Well, if I take a look at how many times we had a recombinant crossing over within our chromosomes in prophase 1 of meiosis, that happened 1,000 times. And so 1,000 is 10% of 10,000, which means that the recombination, the crossing over between these two genes, occurs 10% of the time. And so when I look at an actual chromosome, I can map out the two genes. So here's the gene for eye color, here's the gene for wing shape. Now, they are an arbitrary unit of 10 map units away from each other on a chromosome. So they're fairly close together because they don't cross over very often. Remember, the larger our recombination value, the farther away those genes are going to be. The farther away they are, the more chances they have to actually cross over during that prophase 1 of meiosis. So I hope this helped you to understand how we can use our knowledge of inheritance patterns, linked genes, chi-square, to form conclusions based upon experimental data from actual genetic crosses. Hope you learned something. Thanks.